Good afternoon, everyone. Welcome to our webinar. We will wait for a few minutes for other participants to join. Let's start. Christian Biosystems, established in 2003, is a biotech firm based in Mumbai, focuses on understanding our customers' projects from a technical standpoint to provide cutting-edge solutions to scientists to support their research. We have strong background and expertise in immunology and working with biopharma, CROs, vaccine and academia at pan-India level. Hi, I am Dr. Anisha Polle-Shikdar, Product Manager for Cell and Gene Therapy at Christian. I hold a PhD in cardiovascular biology and have a few years of industry experience. Before joining Christian, I worked as a research scientist focusing on T-cell expansion projects. At Christian, I manage the cell and gene therapy portfolio, including starting materials, viral vectors, and related molecular biology products with a focus on advanced therapeutics. Progen, based in Heidelberg, Germany, a well-established and reliable producer of highly characterized antibodies and standards supporting cell and gene therapy research and development. The range of AAV products, including full and empty capsid standards, enables accurate characterization and quality control across all AAV serotypes. We have Mr. Joshua Bal and Ms. Jacinta Victoria with us today to give us insight on ensuring AV characterization and comparability using reliable AV reference material. Mr. Joshua Bal holds a master's in molecular cell biology and have several years of hands-on industry experience. Before joining Progen as a product manager, he worked 4 years as an ecotoxicologist specializing in plant protection product registration. At Progen, Joshua oversees a broad portfolio of antibodies and related products focusing on advancing gene therapy tools and standards. As we move ahead for the webinar, I request everyone to be on mute and you can drop your questions in the chat drop box. We will address the same during our Q&A session. And now, I would like to hand over Mr. Joshua Bal to continue with his presentation. Please.
Yeah, thank you for the very nice introduction. So, and thank you for everybody joining and listening to me today. So I got the pleasure presenting you some of our data today. So today we are talking about how to ensure AAV characterization and comparability by using reliable AAV reference material. So first of all, since we are talking about AAV today, I just want to briefly give you some facts about AAV, sorry. So it's adeno-associated virus. It's a non-enveloped capsid with a double-stranded DNA genome with a size of 4.7 kilobases.
Joshua, you're not sharing the slides.
Oh, sorry. Oh, thank you. I'm sorry, hang on a moment. So now you should see everything. Is this correct?
Not yet.
Is it shared?
Not for me. I'm not sure about Anisha. I can't see it yet.
I can see.
Sorry? Is it working now?
Yes, yes.
Okay,
Yes.
I'm really sorry for the technical issues.
Okay.
So once again, back to AAV. So I think you saw the slide before in another kind of view. But yeah, this is the AAV virus. And actually, there's also different serotypes available. So ranging from AAV 1 to 12. and it's a non-pathogenic virus, so it only causes a mild immune response in the patients, and this actually makes it an ideal candidate for the gene therapy. And what is also really specific about AAV, I just told you that there's different serotypes ranging from serotype 1 to 12, so these are the really common ones. And what is really cool about the serotypes, they have a tissue tropism. So this means several serotypes are specific for specific tissues within the human body or organs, let's say it this way. And this can be really handy when engineering gene therapies, which should go into a specific place in the human body. All right, so this was it about AAV, and today our main focus is the AAV capsid standards. And first of all, we need to know why AAV capsid standards are actually essential. So you need standards to provide consistency and comparability, for example, between different batches, labs, and manufacturers, but also at the end to evaluate the purity, integrity, and potency of your AAV preparation. So therefore, standards are also important in the analytical method and development validation process, as well as in the whole manufacturing process, especially for the quality control. And it's also a regulatory requirement by the authorities. So since we talked about the regulatory requirements, one of them is to determine or to watch the full empty ratio in your final product. So what does full empty actually mean? So we have AAV capsids, which are considered full. So these are capsids which carry your genetic payload that you desire, that you at the end, for example, want to deliver into your patient. And there's also empty capsids. So capsids which do not contain any genetic material. And why is this important? So for example, for the therapeutic efficacy. So if you want to have your gene therapy product to be efficient, you want to deliver, of course, the full particles because they contain your genetic therapy. And you want to avoid the empty ones due to immune, sorry, I just can't talk anymore. Because empty ones can cause an immune response of your body. So I talked to you before that AV in general only causes a really mild immune response, but basically every particle that you bring into your body can trigger the immune system. So therefore you want to avoid empty ones and only want to deliver full particles because they are the ones which cause the gene therapy. Therefore, there's also regulatory requirements by the FDA. So the FDA wants you also at the end to quantify your full versus empty ratio due to the reasons given above. And so therefore, you also need to monitor the full empty ratio during the manufacturing process. And therefore, standards are really essential. So having full and empty standard material can be really handy, especially during the manufacturing process, for example, as a reference. All right, so talking about standards in more detail, I will just shortly give you some introduction into the history of AV standard capsids. So, for example, there is international reference material, namely AV2 and AV8 by the ATCC. So this is the American Type Culture Collection, which is a global non-profit organization that offers standard material. And this is a really well-known material because it's really highly quality, it's reproducible, and generally really well characterized and validated. And that's the reason why it's internationally considered as the gold standard for AVs. And due to these reasons, we at ProGen, we decided, okay, we want to characterize our internal gold standards, also other serotypes except of AV2 and 8. We want to characterize them the same way as these ATCC standards have been characterized. So therefore, we do, for example, digital PCR for viral genome titer determination, silver staining to determine the purity, electron microscopy for the full empty determination. But nowadays, there's of course a lot of orthogonal methods except the ones listed here. And therefore, it's also important for us to know, okay, do we get the same data that we obtained with these methods listed here with other methods as well, with other orthogonal methods? And therefore, we looked at one example, in this case, the full empty ratio. So the full empties, I told you a few slides before, you should know at this point what I'm talking about. Okay, so here we compared electron microscopy, so EM, with analytical ultracentrifugation, AUC. So if you remember the slide before, EM is the method that was used to determine the gold or to characterize the gold standard material, whereas AUC nowadays is the gold standard method actually for determining the full empty ratio. And we can see on the right-hand side that the filling rate of the capsid standard material that we characterized here is almost identical when using both methods. So we see a really nice correlation between the methods. Then we compared mass photometry, MP, so that's something Jacinta from Refine will talk about in the second part of the webinar in more detail, together with CDMS, and we also see that those two methods, they align really well with the filling rates determined via the other methods before. Then we have the oldie but goldie ELISA, so really well known and established method in the market. So we determined ELISA in digital PCR. The filling rate is a little bit lower with the ELISA, but overall, if we look at the filling rates of all the different methods, we can see that they align really well with each other. So to conclude on the characterization of the filled capsid standard material, we can say that standard materials in generally need to be highly characterized due to the reasons I showed you before, and therefore the help of gold standards can be really handy. Filled AAV capsid standards can also be used to determine the full empty characterization of your final product. So that's something we saw on the slides before. And also the filling rate is really comparable between the methods for our protein standards. Okay, so just a very short overview of how we nowadays characterize our filled capsid material approach. So the titer is determined by our ProGen ELISA coupled with digital PCR. The full empty ratio we determine with CDMS. The purity is analyzed by stain-free Western plotting. And the endotoxin levels are measured via spectrophometric measurements. And the aggregation is determined via DLS. And here a short overview of how our quality attributes look like. So the titer should always be higher than 1 E plus 12 viral vector genomes per milliliter, excuse me. The full empty ratio should always be more than 70% fulls. The purity should be higher, 95%, and the endotoxin levels should be, of course, really low as well. And we can see on the right-hand side that we offer serotypes, like a lot of different serotypes. So there's standard material for almost all the common serotypes. All right, so this was it about the first part of my talk. And the second part will be focusing on the capsid titer determination and also how standards come into place here. So therefore, we first need to talk about what is capsid titer determination and what are current challenges. So in the table here, we see different challenges that we have nowadays with titer determination, and we also see the features that the methods require to handle or to target these challenges. We don't need to go into detail here, but one of them is the comparability between methods. Because, as mentioned before, we nowadays have a lot of different methods, which we can see in the slide here. So, for example, for titer determination in general, there is classical assays such as the ELISA. The ELISA is used for the total determining the total capsid titer. The qPCR is used for determining the genomic titer, and the AUC is used for determining the full empty ratio. But nowadays, there's also a lot of orthogonal methods. This includes mass spectrometry, mass photometry, as well as SEC-miles, for example, but at the end, they all give you a final product result. And therefore, we wanted to know, okay, how is the comparability between all these different methods when it comes to capsid titer determination? So that's why we were running a small experiment or a small study together with a lot of different partners that I will be mentioning in more detail at the end. But what is important for now, we used our emptied capsid standard material from ProGen and sent it to our partners as a reference. And we also sent a material with a blinded titer. So the titer should be determined via the different methods. So therefore, we used AAV2, 5, 8, and 9. And we used the methods displayed here, but I'm going to talk about the methods in more detail in just a few seconds. What important for now is that we used immunoassays, which include ELISA, CE, and BLI, and biophysical assays, which include SEC-MILES and DLS. Important to know is that immunoassays, they use standards as a reference, whereas biophysical assays, they give you absolute values, so they don't require any standards. And really important for the following slides is also the table on the right-hand side. So here we check for the recovery rates that are obtained with the orthogonal methods. So when measuring data with these orthogonal methods compared to the ELISA. So the ELISA will always be the original data we are comparing to, okay? So the recovery rates always refer to the ELISA data. And here are different ranges that we consider as accepted or rejected. So for example, a range of 20 plus and minus 20% above or below the 100% are considered as accepted, whereas minus or plus 30% are considered as rejected. Let's start with the ELISA first, because as I just told you, this will be the method everything will be referring to. So why is ELISA still a popular choice? Some people might think it's really old school, but it's really well established in the market because it's really accurate due to comprehensively characterized standards. It's really reliable due to low assay variances, and it gives you a good comparability between interlab studies, and it's really suitable. And this is one of the highlights for an ELISA kit. You can also handle crude samples compared to other methods where you need purified material to determine your titer. And what is also, of course, really handy, especially for small companies or academics, it's really low cost. So you don't need any special devices. You only need like a plate reader. I mean, this is something nowadays many labs have already available. So you only need the plate reader to get your results. And if you're still thinking in a big scale, you can also automate the ELISA by using additional devices such as, for example, Dynix. So it gives a really precise and reproducible quantification of intact AVs. So what is also really interesting to know is that the ATCC standard material we are talking about, so this gold standard material, was also characterized by using ELISAs. And also it's really a trusted method in really a lot of different companies, and also for the validation of other methods such as SEC-MILES or BLI. All right, talking about the first orthogonal methods. So this is biolayer interferometry, BLI. So what BLI is or does is it measures the change in wavelength of reflected beam due to the capsid binding in comparison to a standard. So we see the BLI sensors on the right-hand side. So the left one, there is no binding of any capsid. On the right-hand side, you see these purple, blue little dots, which represent the capsid in this case. And when a capsid is binding, the length of the wavelength is changing, and so you can measure actually your titer. This will then be also visualized in the graph on the left-hand side. And the most important part is the data obtained with this method, which is displayed in the table on the right. So the table on the right will be important for the next slides because this will be presented for all the different methods. So in the first row, we always see the ELISA data for the different serotypes. In the second row, there will be the respective method presented, in this case, the BLI. And then we see the CV values, which are, of course, really interesting for all of us. And then the most important part will be the titer recovery. So how is the recovery compared to the ELISA data? And in this specific case, we see that the recovery is really nice for AAV2, for example, as well as 8 and 9, because remember, everything in a range plus minus 20%, so 80% up to 120% is considered as a good recovery. Whereas, for example, for AAV5, the titer is a little bit underdetermined compared to the ELISA data. Okay, talking about the next method is CE. So CE is in principle a very precise Western blot in a capillary. So here the amounts of the 3 VP proteins that are contained in an AAV particle of an unknown sample are compared to the protein amounts of a known reference standard. In this case, a reference standard of a third party has been used. This will be important in a second when I talk about the results. So we look at the CV values, they look quite okay. But if we now look at the recovery rates, we see, for example, for AV2, the titer is really underestimated. And also for AV5, it's underestimated. And we might see these differences between CE and ELISA due to the different standards used. So here it's also a good example why the use of the same standards between methods is important to really compare methods. Now we come to size exclusion chromatography coupled with multi-angle light scattering, short sag miles. So this is now a biophysical method. So remember, I told you at the beginning, biophysical methods, they don't need reference standards to function. And how this specific method works is you first have a size exclusion. So different particle sizes are running through the size exclusion, whereas big particles run through the column faster. This you can also see on the graph on the left. So for example, your AAV aggregates, which form the biggest particles, they come through the column first, whereas monomers, which are the smallest particles, they come last. And after the size exclusion, there is a multi-angle light scattering process going on. So light is scattered through different angles. So this is displayed in the photo on the right. And then there is complex calculations going on at the end, giving you your results. So there's definitely technical people which can talk about more detail here, but for us important is the final result. So we see the CV values, they are really low. So this is quite remarkable here. And the titer recovery was also quite okay, except for AV2. And maybe for AV2, which is important to mention at this point, AV2 is really well known in the field to be prone for aggregation. So it really tends to form aggregates. So aggregation might have an impact on the measurements here. The last method is dynamic light scattering. So what it does is measures scattered light, which results from particles within a solution. So if you have a solution, large particles move slower in the solution than small ones. And the method basically measures the size and distribution of the particles in the solution. And then the corresponding intensity leads to determination of the number of particles within the solution. And this number of particles then, of course, give you the titer. And you can also see that CV values here are really, really high compared to everything we've seen before. And also the recovery rates are really low. So that's the reason why we also here in this case had a recovery range. So we checked also for the recovery range, not only the mean recovery. And the high or the significance difference between these methods could be like DLS and the ELISA, that the DLS is basically measuring all the particles in the solution. So the DLS can only differentiate by the size it measures, but not by the particles. So it could be a particle which is an AV particle. At the same time, you have another particle in your solution which does not belong to AV, but got the same size. And then it gives you a false result. And that's also one of the reasons you can't use this method for crude samples. So you need really highly purified material. All right, so now we come already to the conclusion. So everything is basically what we saw on the last slide is concluded in this picture here. So you have the capsid titer recovery on the left-hand side, the different serotypes below here. So if we look at AV2, or let's say let's first define the line. So you see this blue line in the 100% range. So that's basically our ELISA result. So this is the reference for everything else. And then the green box indicates the accepted range where we accept the measurements or the recovery rates compared to the ELISA. And if we now look at AV2, we see that basically 3 out of 5 methods underestimate the titer of AV2, whereas it's better for the other serotypes. As I said, aggregation might be a factor here. So there are several factors influencing the measurement in general, and one might be the serotype-specific factor in the specific case of AV2. And if we now look at the different methods, for example, we see that DLS for 3 out of the 4 serotypes, so this is the light green dots, was always in a rejected range. So the DLS was really the most unprecise method compared to the other ones due to the reasons I mentioned before with the particles in the solution. And what is really interesting to know is that, or to see, is that the ELISA results and the BLI results, they correlate really well with each other. And this could be due to the same standards being used in the method. So remember, BLI and ELISA both use the protein standards for the same reference material. And we see that if you have the same reference material, you also could really get really nice correlating results. All right, so just to sum it up, since we used our empty capsid standard material here for this experiments, I just want to also indicate how we determine the different characters of our empty material. So basically, it all remains the same to the filled capsids, except of the full empty ratio determination. We use UV this here, except of CDMS, or instead of CDMS. And here again, the quality attributes for our empty capsids. So we always want to have a titer higher than 5e plus 12 capsids per mil. And we want to have an empty ratio higher than 90%. And the purity rate, again, as with the filled capsids, higher than 95%. And also endotoxin levels, again, below 1 EU per mil. And again, as we have seen in the filled capsids as well, we have this available for several serotypes. All right, so at this point, I really want to thank everybody who was involved in obtaining this data, and again, highlighting also Krishkin for giving us the opportunity today for presenting. And sorry, I also want to excuse again for this technical struggle at the beginning with the screen, but I hope everybody was able to follow, and I'm happy to take your questions at the end. And yeah, thank you most importantly for everybody who was joining and listening, and thank you very much for your attention.
Thank you, Joshua, for the nice and insightful presentation. Now, allow me to introduce Jacinta. Jacinta Victoria is a strategic marketing and analytical leader with over a decade of experience in the biotechnology and gene therapy sectors. As the market development manager at Refin Limited in Oxford, UK, she leads initiatives, campaigns to drive the adoption of mass photometry across critical applications in cell and gene therapy, including AAV, LVV, adenovirus, and RNA-based platforms. To continue with her presentation, I request the same to everyone. Be on mute and you can drop your questions in chat drop box. We will address the same during the Q&A session. Jacinta, over to you. Yeah.
Thank you, Anisha. Can you see my slides?
Yes, yes.
Perfect. So good day, all. Like Anisha introduced, I'm Jacinta Victoria, Market Development Manager for the CGT space with Refine. And for today's presentation, I would like to talk about quantification of the heterogeneous AAV capsids using our technology mass photometry and how standards, as in reference standards, play a significant role in it. So as part of the presentation, I would like to touch on the significance of the fit-for-purpose analytics. Again, the significance of standards is part of it, to introduce our new technology and how our technology performs in that space. So before I dive into fit-for-purpose analytics, I wanted to broadly touch on the significance of CQAs. So CQAs, or critical quality attributes, are defined by regulatory agencies, and it's key to monitor them because they help ensure that your product is safe, is efficacious, has high quality, and also meets regulatory standards. Now, from previous experience, we now know that failure to effectively manage your critical quality attributes for the product can lead to clinical trial failures and also safety concerns, including death. So here are some of the risks that are highlighted that have been taken from case studies of analogous therapeutic molecules, and they include modified biodistribution of the product, reduction or compromise of the therapeutic function of your desired capsid, and finally, heightened safety concerns due to the presence of impurities and/or incorrect dosing. So now this gives you a broad overview of the critical quality attributes for AAV products. So as you can see, they are split as content, identity, safety, potency, physical, and purity. And each of those boxes have a few bullets below. Those are the attributes that fall under each of those qualities. And as we all know, each of these attributes will have to be measured using a reliable analytical tool. And for each of these analytical tools, which is what the content of this presentation is, you have a driving factor, which is the key factor, which is having reliable reference standards. So now let's talk about the significance that the reference standard brings to enable robust analytics. And here are some of the key points which highlights that significance. So again, Joshua gave a beautiful presentation touching on all of these concepts, but just to bring the message home, I'm going to sort of iterate on it. So as we know, reference standards, they set your benchmark. So they help you understand that the assay you've developed or the assay that you're performing is actually, it's doing what it's intended to do. So in this case, let's say for an ELISA, it's actually measuring the intended analyte AAV capsid. And also it helps you understand that the measurement is done accurately, reliably, and consistently across labs and across time. Also, when you think about collaborative research activities happening across labs, across agencies even, you want to have a comparator tool. In this case, your comparator molecule is your reference standard, because this helps understand or help you achieve that reproducibility and the comparability that's happening across labs, and also helps you track the traceability of the process and the batches. So when you think about regulatory considerations for methods, every assay, every analytical assay goes through two checkpoints, which is assay qualification and assay validation. And for this to be achieved, you have to have acceptable performance characteristics and limitations set for each method. And these limitations or specifications are set using reference standards. So not just that, your reference standards also help understand or track the performance of your assay across your qualification process and the validation process. Your reference standard also acts as a monitoring tool. So it helps you understand the drifts in the assays, and it helps you ensure or helps you understand that your method, the data that you're obtaining from the method is reliable and accurate. So this is key because during process development or process optimization, there is a lot of process changes happening which can impact your product. Now that your reference standard enables robust analytics, now any changes in the trends help us understand that that is the true change coming from the process on the product. And finally, because reference standards are able to demonstrate the robustness and reliability of the method, this speeds up the approval process of the regulatory agencies because we are compliant with it. And finally, it helps achieve that industry consistency across the industry. So now going back to the significance of fit-for-purpose analytics. So for scope of this presentation, I'm going to only talk about the product impurity, which is empty-full analysis. So again, Joshua gave a very comprehensive introduction for AAVs, but I'm just going to touch on a few factors again as a background. So as we know from all of the previous research, there are 4 AAV populations. So there's your empty capsids, your full capsids, your partial capsids, and your overfull populations. Your desired capsids are your full capsids containing your intact transgene. So when you think of your ideal AAV product, you should have enriched amount of your full AAV capsids. But unfortunately, your impurities, which are your empties, partials, and overfulls, also co-purify with your full capsids. So now, with all of these impurities present in your sample, it can impact the potency of your full capsid, can have increased immunogenicity, and also can increase the cost because now you're trying to remove these impurities from your product at a later stage in the process, so your product is regulatory compliant. So because of all of this, now let's answer why is it critical to have the right tool for empty-full analysis? So you want to have a product which, like I said, should be highly potent, has less immunogenicity, and is safe and efficacious. So to understand that or to achieve that, you want to have precise quantification of that product to help understand what needs to be changed in the process to achieve that. So your tool should give you that accurate and reliable quantification to make all of the desired changes. And now, as we touched on in the previous slides, your reliable standards help enable robust analytics. So now, because of that, you're able to understand changes in the trend, which reflects the true impact of your process on the quality of your product, safety of your product, and efficacy of your product. And finally, because you're able to achieve that control over your process at very early stage in the process, you're able to obtain high yields of your product with no compromise on the quality, safety, or efficacy of the product. So now finally, that is why we want to have, or why we want to choose the right tool. But what should that tool look like? So your tool should give you accurate and reliable analysis of your empty and full populations, and this will be achieved with established reference standards for this method. And also it should reliably quantify your newer populations, which are your partials and overfull particles. But all should be done in few minutes with low sample volume and at very low operational cost. So one key factor here is that, as Joshua touched on, AAVs have a number of serotypes. So it's ideal that your right analytical tool should be serotype agnostic, so it can be actually adapted across the different serotypes that your company is working on. And finally, because we spoke about process impact on samples or process impact on quality of product and process control, we want this right analytical tool to also be a process monitoring tool. So it can be adapted from very early stage in the process up to the later stages. So we want to introduce our technology, which is mass photometry, because it checks all of these ideals for being a right analytical solution. So mass photometry, as its name goes, it measures the mass of your single particles in solution using light. So as you can see in the image in the center, you have your particles in solution on a glass slide. And when illuminated with a laser, all of your particles scatter light, forming a contrast pattern. So here, in this case, your contrast pattern is actually a circular dot. This contrast pattern or circular dot is proportional to the mass or density of your particle. So as you can see here, darker the dot, heavier your particle. So now what the software does is it takes this contrast information from each of these particles and converts it into mass using a standard, again highlighting the significance of standards for every robust analytical tool. So based on the shift in mass, so as we know, we all know the mass of empty AAV capsids. The full AAV capsid mass is defined based on the length of the insert. So using this information, now the software differentiates the different populations. So they segregate, the populations get segregated based on the difference in mass. Because the empties have a set mass, as I mentioned, your fulls have a higher mass than the empties because of the insert. And finally, the population that falls between the two is your partials. So this is the histogram generated on the SAMX, which is a dedicated mass photometer for AAV characterization. So now let's talk about the performance of mass photometry in the AAV space. So before we go into that, let's again touch on standards and what are the consequences of not having good standards. So as we spoke about previously, we want to have good standards for tracking a number of things and for enabling that robust analytics. But as we know, cell and gene therapy, again, is an evolving field, and there aren't many reliable gene therapy standards that can facilitate good tracking across labs and good tracking across manufacturing facilities as well, which can actually set industry consistency. So having appropriate gene therapy standards is key because it helps facilitate your process development, process validation, and also keeps consistency or track of consistency across the performance of the process across the different batches. So here is an example of an AAV8 full standard that was measured on the mass photometry. So as you can see, this is a full standard. And again, based on how I explained the gating strategy previously, this standard is a reliable full standard, again, confirmed by the mass and the population that's gated in that space. Again, to reiterate, you want these good standards to be available for every analytical method because especially in the GMP space, for the QC environment, it helps performance, as in consistency of performance across every testing phase. It helps with the regulatory consideration of having your methods go through assay qualification and validation. It helps with that and also helps you track the performance across, and also it helps you track the performance of process across batches. So that is why we want to have reliable gene therapy standards which enable robust analytics, which come as a combination, which can actually bring industry consistency and also help enable high quality products. So now let's talk about benchmarking. So mass photometry has been available in the industry for a few years now, but as Joshua mentioned, AUC is like the industry standard analytical ultracentrifugation for measurement of the different AAV populations. So this has been taken from a publication by our industrial partner Takeda, where they've used mass photometry, which is the purple bar, against AUC, which is the blue bar, and they've measured 3 different serotypes. Going from left to right, you have AAV5, AAV8, and AAV9. As you can see from each mass photometer has actually measured 4 different populations, which are your empties, partially filled, full, and overfull, all in alignment with AUC. So this helps us conclude that mass photometry is serotype agnostic, as we highlighted in the previous slide, and also it reliably measures your different AAV populations, all in alignment with the current industry standard analytical ultracentrifugation. So as we touched on that, right, like an ideal point for the right analytical tool, you want your tool, especially in the AAV space, to be serotype agnostic because of the number of AAV serotypes that the industry is working with. So to understand the serotype agnosticity of mass photometer, we did this experiment internally where we took 4 different AAV serotypes and used the same workflow with no changes to the workflow and measured it on mass photometer. Based on the results, we understood that there was no serotype-specific bias coming from the measurements, therefore helping us conclude that mass photometry is serotype agnostic. Now, to further add value to that, we collaborated with our industrial partner, who is the HEC lab at the Utrecht University. So they specialize in CDMS, which is charge detection mass spectrometry, which is another tool used in the AAV space for measurement of empty-full analysis. So what they did was they took few AAV8 and AAV5 samples and measured it on a mass photometer, which is the center histogram colored in purple. And they took one of the AAV8 samples and measured it on CDMS. And when the mass of the population was compared between the two, they were very closely aligned, further helping us conclude that SUMX is serotype agnostic and aligns reliably with the industrial standard AUC and charge detection mass spectrometry. So this is to further highlight the position mass photometry has in the industry. So US Pharmacopoeia has recently included mass photometry in their gene therapy analytical guide for AAV product and process characterization and also for QC release and stability. A snippet of the website has been shown on the left. Mass photometry has also been included in the General Chapter 1067, which outlines the guide for AAV manufacturing and QC. So this is critical because now USP Pharmacopoeia has very well featured mass photometry on their forum, helping or reinforcing that mass photometry is a reliable tool to be adapted for both characterization and QC release in all environments. So again, as we previously touched, one other factor for being a right analytical tool was process monitoring. Because as we said, to ensure that your product is of highest quality, you want to have real-time analysis of or understand the real-time impact of your process on your product, for which you want an analytical tool that's fit for all of the process changes and is not impacted by impurities or by any sort of process buffers or components that can be during process optimization and development. So this is an example. So this experiment was conducted in collaboration with our industrial partner Genathon. So here we wanted to understand if mass photometer can be adapted in early stage in the process, which is upstream, and also understand if it actually carries a significance of being adapted in that space. So for that, Genathon used a simple cleanup process which they've developed, which is pretty much a plate which contains resins ligated to AAVX antibodies. So anyone in the AAV space would know that these AAVX antibodies are used for purification during the scale-up process on the affinity chromatography. So this takes roughly about 3 hours. So the harvest material from either the shake flask or bioreactor was run through this plate, and samples pre and post purification was measured on the mass photometer, the histograms shown on the right. So as you can imagine, your sample coming from the bioreactor One has a number of impurities coming from the host cell, the plasmids that are remnant during transfection, and also all of the media components and protein components that were added to feed the cells. Apart from that, at that stage of the process, you have very limited amount or very diluted amount of your product in there. In our case, your AAV capsids. So all analytical tools, irrespective of the scope, get impacted because of the presence of impurities, presence of other host cell proteins, host cell impurities, and also because of the diluted presence of your product. So here, the clarified lysate, which is your pre-purified sample, as you can see, had an increased amount of impurities, the left, and then nothing really or no definition of your AAV capsids as seen in the A graph. When you move on to the purified version of your sample, so here we've shown example from two different serotypes, AV8 and 9. Post-purification, you see a depletion of the impurities and enrichment of your different capsids. Again, why is this important? Because at very early stage in the process, you're making very critical decisions, as in your transfection optimization, which cell line to go for, your plasmid construct design even. So to understand the true purity profile of your product is very important, because now you're able to make data-driven decisions and have better control of your process at the early stage. So therefore, you can drive high-quality product at the later stage in the process. So now that we've shown that mass photometry can be adapted in early stage, but also the significance of having robust analytical tools to measure true purity profiles, now we wanted to understand if it can serve that purpose in downstream phase of the process as well. For that, we collaborated with our industrial partner who took the AAV8 vector and purified it using two different manufacturing protocols, A and B. And the sample was measured on the mass photometer to obtain the purity profile. So the histograms here show that you have a better purity profile of your product with manufacturing A protocol, because as you can see with manufacturing B protocol, you have an increase in the number of partial capsids and a decreased amount of your desired capsid, which is your full capsid. This was also evident from the poor transduction efficiency coming from the cell-based assay. Again, data is not shown here. So here again, we highlighted that you want to have robust manufacturing process to obtain high quality data, but also to have reliable standards which enable robust analytics, which can actually help you understand the true impact of the process on the product. So to sum it up together, yes, you want robust manufacturing process, you want reliable standards, and finally, you want robust analytics to help ensure that you have good or high quality product at the very end of the manufacturing phase. So SAMX has proven to be a good process monitoring tool, going from very early stage in the process to the later stages in process. So as we know in the industry, this is just an education piece. As we know in the industry, we have three different methods, which is DLS. Again, Joshua touched on it. It's the dynamic light scattering tool, which uses Brownian motion as the principle. We have PCR and ELISA. So these are three tools which are very widely used in the gene therapy space for titer determination, for aggregate analysis, but also for empty full analysis. So here we wanted to understand if that is true and what the impact is if we were to use these methods for in-process samples. So Joshua very clearly explained the reliability of the measurements for clean or final drug products, which are your gene therapy standards. Whereas here we wanted to understand if it can be adapted for process monitoring. So here we took three different samples during the manufacturing process, which is capture, polish empty fraction, and polish full fraction, and measured it on mass photometry, the orange bar, DLS, the dark blue, and the ratio of PCR ELISA, which is the light blue. So mass photometry was used as the baseline because we've actually shown data that it's comparable to the industry standards, AUC and CDMS. So the dPCR ELISA, the light blue bar, in all of the cases overestimated when compared to NP. So this can be attributed to, if you think about it, as two different methods. They come with inherent variability, and when taken as a ratio, the error compounds. So that is why you have that overestimation. Whereas DLS, on the other hand, underestimated in comparison to mass photometry. Again, this can be attributed to its sensitivity or requirement to have high concentration of your capsids to provide reliable data, and also the negative impact coming from the presence of impurities. So again, although your DLS, PCR, and ELISA does have a reliable position in the gene therapy analytical toolbox for titer and aggregate measurements, it is very unreliable to be used for empty full analysis. So to conclude, SAMX is a reliable tool for empty full or for measurement of the different AAV populations and also to bring the message home, irrespective of the method, to have reliable reference standards that can actually ensure accuracy and also regulatory compliance. So now to sum everything up, obviously we've shown previously the capability of SAMX or the mass photometer to measure heterogeneous populations. But in this case, we wanted to understand the reliability of the measurement across the whole testing range. For this experiment, we took empty and full standards and mixed them in different ratios going from 0% to 95% full and measured it in triplicate. So the histogram on the left, as you can see, the titration of the empties and fulls going across. So when the measurements were plotted on a graph, which is plot against predicted loading to measured by the mass photometer, you can see a best correlation fit and also high reproducibility from the diminished error bars from each of those data points. So again, this helps us bring the message home that SAMX is reliable for measurement of heterogeneous AAV populations across a wide analytical testing range. So to further bring confidence into the reproducibility of the method, we measured a sample 18 times over 3 different operators. And the mass measurement and the empty full analysis from each of those measurements were summed together to get the variation profile, which is the percentage coefficient of variance. As you can see, the highest coefficient of variance obtained was 5%. Again helping us build confidence that SAMX is reliable and also reproducible in its measurements. So finally, to summarize, yes, SAMX is a comprehensive AAV characterization tool. It is a single particle serotype agnostic tool which can help measure your different AAV populations all in alignment with AUC and CDMS. Again, to very strongly highlight, this robustness is actually enabled by having reliable analytical reference standards. So because of its intuitive workflow and because of the quick run and the analysis, which is less than 5 minutes per sample, you're able to train it out very easily to new personnel and also can be adapted into every environment. With the release of the GMP software, it can now be used both in PD space for process monitoring, but also in GMP for QC release. So finally, yes, as I said, you need robust analytical tools and also reliable reference standards which showcase the robustness of whichever analytical tool that you're using. So to introduce our portfolio, so we have 2 mass photometers, the SAMX, which is dedicated for AAVs, which we spoke about in large detail previously. 2MP is another mass photometer, which has been designed for proteins, antibodies, nucleic acids. So both of these tools have both manual and automated workflows and cover different mass ranges as mentioned here. A new inclusion to the portfolio is the Characterization, which is also called macro mass photometry, because it not only measures the mass as the mass photometer, but it also helps define the size of larger viral vectors, such as adenovirus, LVVs, VLPs, and also LNPs, in the size range of 40 to 150 nanometer. So thank you for listening in, and thank you again for this opportunity to present here, offered by Christian. I'm happy to take any questions.
Thank you, Jacinta, for the wonderful presentation. Now we'll have a small Q&A session. And in case if you have any questions, you can drop in our chat box. Okay. We have 2 questions for Joshua. You showed us many different methods for determining the capsid titer. Which method would you personally recommend for AAV titer determination?
Yeah, that's actually a really good question, and it's always also depending on what you're looking at. For example, if you work a lot with crude samples, crude lysates instead of purified materials. So if you want to have, for example, an in-process control of your titer during manufacturing, I think ELISA would be the method to go to because it gives you just really a flexibility in determining your titer. Whereas, for example, the dynamic light scattering is maybe not as highly recommended due to the reasons I also talked about in my presentation. So there is not really a differentiation between the particles that are measured, especially if you're working with crude lysates. So I hope that solves the question.
Thank you, Joshua. And next question is also for Joshua. How is the lot-to-lot reproducibility of the capsid filling grade of your AAV reference standards?
Also a good question. I mean, it's also a question that reaches us a lot. Of course, there's always a little bit of variability from lot to lot. This is biology, of course. But concerning our full capsid standards, we always try to have a purity above 70%. So this is something we definitely guarantee that the purity is higher than 70%. We sometimes even reach 80 to 90%. So for full capsids, that's actually really good value.
Okay. Thank you all for attending our webinar today. And we are grateful for your participation and hope you found the session informative and engaging. We appreciate you taking the time out of your busy schedules to join us. Your engagement and thoughtful questions contributed greatly to the success of the webinar. A special thank you to Mr. Joshua Bal and Ms. Jacinta Victoria for sharing their expertise and insights on AAV tools and analytics. The recorded session will be sent to you, and keep an eye out for our email on promotional offers only for the registered participants. Thank you.
Thank you. Have a great day.
Thank you. Bye.
Thank you, Jacinta.